Hello everyone, in this tutorial we will learn to investigate genetic cause of a rare monogenic disease. As part of the investigation, we will collect relevant information about the monogenic disease from publicly available databases and apply this knowledge for identifying putative causative variant from a given VCF file of an individual. As a use case for this purpose, we will investigate genetic cause of cystic fibrosis, an inherited disorder that causes severe damage to the lungs, digestive system, and other organs in the body. To gather the information about this disease, let's explore the Genetic Home Reference Database maintained by National Library of Medicine that provides consumer-friendly information about the effects of genetic variation on human health. Querying this database with cystic fibrosis returns a web page with all relevant information about disease's description, prevalence or frequency in human population, causes, and inheritance pattern. Let's explore this page by asking clinically relevant questions for investigation. Question 1. What is the prevalence of cystic fibrosis? Clicking on the Frequency tab displays this information. As written, this disease is common within white population in the United States compared to other ethnic groups. The prevalence in white newborn is 1 in 2500 to 3500. However, in African Americans, it's rare, that is 1 in 17,000, and in Asian American, it's very rare, that is 1 in 31,000. Question 2. What are the genes associated with cystic fibrosis? Clicking on the Causes tab displays that CFTR gene is associated with this disease. Question 3. What is the inheritance pattern of cystic fibrosis? Clicking on the Inheritance Pattern tab displays that it is inherited in an autosomal recessive pattern, which means that both copies of the gene should carry a pathogenic mutation. This indicates that an affected patient should be either carrying a homozygous causative variant or should have a compound heterozygous causative variants, a case where the two copies of the gene carries two different pathogenic heterozygous variants. Information from these three questions will be later used for identifying the putative causative variants. In the next part of this tutorial, we will investigate a case of identifying putative causative variant in an individual of American Asian origin who is suspected to be suffering from cystic fibrosis. We are given a VCF file containing 129 variants for the individual. The first step in this investigation requires annotating the VCF with genomic region, allele frequency, mutation type, and predicted impact. We will use WANOVER tool for the annotation. As this tool was discussed in the tutorial of the previous module, we will directly analyze the results here. Identifying putative causative variants requires variant prioritization steps to be executed. Let's answer a series of questions as part of the variant prioritization step. Question 1. What is the name of the gene associated with cystic fibrosis? As learned from the Genetic Home Reference Database, CFTR is the gene name. Question 2. What are the variants on the CFTR associated gene? The WANOVER results page provides an user interface with options of filtering variants in genes. Using this feature, we see that there is only one nucleotide variant, that is on chromosome 7 position 117120149, where A is substituted by G, is present in the CFTR gene. Question 3. Is the variant of good quality? To access this information, let's open the VCF file in Excel and look at the filter column. A pass value in this column indicates that the variant is of good quality. Question 4. Is the variant homozygous? Yes, The variant is homozygous as it has genotype 1/1. The recessive pattern of inheritance of cystic fibrosis is represented by homozygous condition of the variant, and therefore one needs to check variant's homozygous state. Question 5. Is the genotype quality of the variant good? Yes, the genotype quality looks good and is above a threshold value of 30 as recommended by the variant calling software. Is the variant a rare or a novel one? Getting back to the W-ANOVA annotation table, according to the 1000 genomes, it is a novel variant, and in EXAC it has a very low minor allele frequency. As the VCF file is of an individual with Asian American origin, where the prevalence of cystic fibrosis is very low, such low minor allele frequency of the variant matches the variant prioritization criteria. Question 7. Is the variant in exonic region? If yes, what type of mutation does it cause in the protein? The func column indicates that it is an exonic variant, and the exonic func column indicates that it causes start-loss mutation, that is, a type of missense mutation in the protein, where the first amino acid, methionine, is substituted by valine. Question 8. Is the variant previously reported as pathogenic in clinical databases? For this, let's go to the ClinVar website. But before that, let's get the RS number for this variant from this page. This variant has dbSNP identifier as RS397508328, and we will use this number to retrieve information about this variant in ClinVar. In the interpretation section of ClinVar, the variant is reported as pathogenic, and the assertion and evidence section indicates that this assignment has been submitted by 4 groups, and one of which is reviewed by expert panel. Question 9. Is the variant predicted deleterious by in silico impact prediction tools? The polyphen2 HDIV pred column shows that the variant is possibly damaging, indicated by letter P, and the SIFT pred column also shows that the variant is damaging, indicated by letter D. To summarize the finding from a very basic analysis, we identified a very rare non-synonymous variant in CFTR gene that has been reported as pathogenic in clinical database and also predicted deleterious by in silico impact prediction methods. This variant could be a putative causative variant in the individual whose VCF file was being analyzed. However, further experimental validations should be performed to verify deleteriousness of such putative causative variants. This brings us to the end of this tutorial, where we learned to investigate genetic cause of monogenic disease using computational tools. Thank you for watching the tutorial.